This is episode 40 of the Immunology Podcast, Bacterial Pathogens with Drs. Kristin Patrick and Robert Watson. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Drs. Kristin Patrick and Robert Watson from Texas A&M University on the podcast here to talk about their research investigating the interface between intracellular bacterial pathogens and the innate immune cells that sense and respond to them. We've also got our usual roundup of recent highlights in immunology news coming up. But first...
You know, Jason, performing multiple rounds of cell isolations can be inefficient sometimes. Thankfully, you can use the new EC250 EC-CEP magnet, where you can scale up your cell isolation and process large volume samples like leukopaks and whole blood, all in a single round of separation. You can obtain highly purified cells from samples up to 225 mils in a single step. Learn more at www.stemcell.com/EC250 magnet.
That's awesome, Brenda. I bet you could go outside and see the daylight sometimes if you used one of these.
I know, right? I have to say, I've never done, I've always did the whole, fecal isolation for PBMCs. I should really check one of these magnets in my future life, because it must be kind of nice to just do it directly from blood.
I remember my first fical. I only had like 2 mils of mouse blood total, and I couldn't see anything.
Oh no, well that's yeah. I mean the big fical, like a 50 ml Buffyco that you get from the blood donation center is very satisfying. It's a very thick, nice layer, easy to pick up, and it's like, yes, I definitely have cells here.
Oh, yeah. I've seen some Buffy coats in patients who had triglycerides in the thousands that are a little frothy.
Ugh, yeah. Well, human serum, compared to mouse serum, I remember it's quite fatty.
Rich. Yeah.
Yeah.
Especially with our diets these days.
Must be bad. Yeah.
But you know what? I bet Neanderthals, they probably didn't have fatty diets. Did you see that the Nobel Prize was on the Neanderthal genetics in medicine?
What a beautiful segue.
I had to do it.
Indeed. It's funny because I will be talking about some old DNA today. Not Neanderthal old, but pretty old. So I think it's very appropriate. So very much congrats. That was for physiology or medicine or chemistry? I forgot.
Physiology of medicine. You're right on both accounts because it's both words.
I was both words. I always thought it's physiology or medicine.
No, it's physiology of medicine.
Okay. I do think there should be a Nobel Prize for biology. Why does it have to be medicine?
Go ask the people who set this up a long time ago.
So what does your... I assume you already had a DNA test. How much percent Neanderthal are you?
I have not had a DNA test because I know what they do with that data, and I have shared it with the larger world. I've only had partial sequencing for genes of Jewish concern. Jewish people have a certain number of genes that are more common among Ashkenazis that can lead to disease when you're that are autosomal recessive. And so I got screened, and since I didn't have any, then my wife did not have to get screened because I was 0 for whatever.
Okay. I thought it was some kind of pedigree thing. It's like, well, this is nowadays, all this matchmaking got very complex.
No. This was, like Tay-Sachs.
Okay.
Those fun things.
Fun. Yeah. All right. Very nice.
So anyway, I know we got papers here. Do you want to start with some ancient DNA and just get that one out of the way?
Yeah, you know what I do? I think this is a very recent paper. Actually, it just came on this 19th of October. And I thought it was very intriguing, very cool. So this is, the paper is called Evolution of Immune Genes is Associated with the Black Death. That's a very interesting name. And the paper basically looks into DNA that came from skeletons recovered from cemeteries right before, kind of during and after one of the larger episodes of bubonic plague in Europe and Asia. So there was the black, what is kind of known as the Black Death, which would happen around between 1346 and 1350, according to I guess that's kind of these years in which bubonic plague really disseminated European, probably also Middle Eastern, Northern African populations. People say up to half of people died, or like at least a third. I think a third is a less controversial number. And I mean, imagine living there. It must have been, oh my God. I when I think about it, I just kind of like my hairs get a little bit, the hairs in my neck get a little bit tense. So for this research, they got material from 206 Asian DNA extracts from people buried in uh, cemeteries in London and around Denmark. And in the case of the London cemeteries, there were very specific dates, so they could really kind of date this DNA from either like before the Black Death. There was one cemetery that was mostly occupied by victims of the Black Death, and then there were posterior things that were people that were buried after, several years after the fact, and they looked into several loci. Either they had some loci that were associated with immunological genes. They had some loci that had been already associated with different disease or immune related conditions, and they had some loci that they kind of had like as a control. They were not expecting major variation. And so they did a lot of kind of analysis of this kind of they compare the prevalence of these different isoforms of the different loci between these different variants of this loci between the different material that obtained. And basically they got a couple of loci, a couple of alleles that were variants that were kind of represented preferentially in one of the groups. And I think the most interesting, the one they had kind of the highest signal from, was this. It was a loci that was near a gene called ERAP2. And so they had a variant that was was associated to a different alternative splicing of this gene. And they see that there's two variants of this gene, and one of them is associated, is much more expressed, is after the Black Death, so as associated with kind of survival to the Black Death. I just need to maybe mention that they kind of really tried to find people that were descendants of people that survived the Black Death, in contrast to people that maybe immigrated from other places after the fact. So they do think that this kind of this different variant is evidence of a selective advantage that is stemming from this variant. And interestingly, so this ERP2 is a aminopeptidase that is associated to trimming peptides for presentation to T cells by MHC class 1 molecules. And in the way they present it, they think that this particular variant that is associated with kind of higher mortality results in a splice variant that has a premature stop codon, which results in kind of the loss of this protein. And then the loss of this protein prevents presentation by macrophages, which are preferentially infected by Yersinia pestis, to be kind of targeted by CD8 cells. And then this allows the Yersinia to infect the macrophages, to be taken to the lymph nodes, and to kind of start replicating uncontrollably, because we do know that lymph node infection is very swollen lymph nodes, and lymph node infection is very typical of bubonic plague. So it was very interesting because they get to this through looking at these variants and how they are represented in different populations. They do some experiments because they find that these different variants are still present in modern people, and so they get their hands on PBMCs derived from 10 individuals, 5 of which are homozygous for the selectively favored allele, and 5 people that are homozygous for the alternative allele. And they also see differences in the virulence, kind of in the in how these cells kind of respond to infection by Yersinia pestis. So I thought it was very interesting. I thought it was very interesting that how they got to this, and I think was also very cool is that this this variant, this ERAP2 variant, is also apparently has been associated as a risk factor for Crohn's disease. So it's kind of gives you this idea of the trade-off between sometimes having a more capable immune system and autoimmunity, and kind of being predisposed for autoimmunity. So I thought that's also very interesting, and so pretty cool in my opinion.
It's interesting how often macrophage uptake of pathogens, which is what they're supposed to do, and then chew it up, is actually the nidus of infection.
Right?
Like, it gets hijacked all the time.
Yeah. What is wrong with these macrophages? Like, come on, guys.
All right. Well, I'm going to talk about an intracellular pathogen. Segue.
Beautiful.
Tuberculosis.
Okay.
The big daddy of all intracellular pathogens.
I mean, another pathogen that hijacks macrophages, no?
Yeah, it is. So this is a bacterial phospholipid phosphatase, inhibits host pyroptosis by hijacking ubiquitin. It is in Science. First author is Kuo-Yao Chai. Last author is Koi-Hu Liu. And it came out October 14th. So, as we're going to hear later on this podcast, I'm a big fan of Gasdermin D as a molecule. This is a protein that forms pores in the membrane and causes a cell death known as pyroposis that's inflammasome mediated. It gets cleaved by caspase 1 to then form pores. These pores release a bunch of cytokines and the cell dies. So it's an inflammatory mediated cell death, and it's super important and critical. And it targets the membrane by the phospholipids present on the inner leaf of the membrane. They did a really sophisticated screen looking for proteins released that are in the tuberculosis genome that have eukaryotic-like motifs. And then they found one that was PTB, also known as RV0153C, very abundantly secreted during infection. And they found that it had a binding domain for phospholipids. And they found that it could target and dephosphorylate host plasma membrane PI4P, so phosphoinositide, and PI45P, PIP2, which then would prevent Gasdermin from going to the membrane, making a pore, and letting the cell die. So they found this out here. So that was the first thing. They found, hey, this has a phosphatase. If you overexpress, if you knock it out, you get more Gasdermin-mediated pyroposis. They found that it had phosphatase activity. They did all this mapping of that, right? But then the story doesn't stop there. That's what is so crazy. And so what they found is that during infection, different mutations had a different effect. And they found in the end that this thing gets ubiquinated. And it has some activity at baseline, but actually what is driving it, because they did all these things where they knocked it out or overexpressed it in a cell, and so you had the native host mechanisms there. They found that in vitro it wasn't working as a phosphatase. And then so they looked so you're not getting phosphatase activity in vitro, which is a giant pain in the butt, right? Kind of ruining the paper. And then they found that it had a ubiquitin binding motif. And they really dived in. And they found that this protein actually is bound to ubiquitin. And then they showed through mutagenesis what the bind domain is, but not ubiquitin-like proteins. And that ubiquitination opens up the active site for function to do its work. And so what happens is this is a protein secreted by TB, which inhibits the normal intracellular pathogen sensing response to lead to pyroposis. If you get rid of it, mice, all the inflammatory markers, everything, they do better in the face of TB if you knock out this protein or the gene. And so you have an intracellular mechanism that's actually a eukaryotic phosphatase. And remember, TB doesn't have PIP, right? It's not part of how they signal. It's only a eukaryote thing. So I thought this was really cool.
So they have it only in order to mess up with their host.
Correct.
Cool. But sorry, I missed one thing. So pyroptosis, so pyroptosis bad for MTB, right?
Correct. It kills the cell, and then MTB, and then it's no longer an intracellular pathogen because it got thermonuked. And they actually see, interestingly, granulomas increase. You have to have the inflammatory cascade for the granulomas, and so only granulomas are present in that. You get more granulomas in the absence. If you turn off Gasdermin, knock it out with TB, you get
more granulomas.
Mice die, but no granulomas because there's no inflammation, right? They don't. Because part of pyroposis is that the pore releases the cytokine.
And so the ubiquitination, does it not get degraded because of the ubiquitin?
Eventually, presumably, they don't go into the degradation of it.
Okay. Another way MTV is just messing with us forever.
Yep.
Nice. Well, an evil bacteria. Before I move on to my paper, I just want to say that I did not mention the authors of the paper I mentioned before. So I just want to give a big shout out to Jennifer Klunk and Taras Bilgalis, who are the first authors of this paper titled Evolution of Immune Genes Associated with Black Death, from the labs of Hendrik Poyner at McMaster University in Ontario and Luis Barreiro at the University of Chicago. So I just wanted to make sure credit is properly given. And that being said, let me take you into a journey into structural biology. Are you ready?
I love structural biology.
I have to say, I appreciate structural biology. I'm not a connoisseur of it, so it did take me a little bit of kind of research, a little bit of a rabbit hole of cryo-EM to kind of understand this paper. So the paper is called Structural Principles of B-cell Antigen Receptor Assembly. It was published in Nature, 13th of October. First authors Ying Dong and Xiong Pi, from the labs of Hao Wu at Harvard Medical School and Michael Reth at University of Freiburg in Germany. And I think this is a very interesting paper because I would say maybe as a non-structural biologist, it's not that easy to understand, but it is very important because this is the first reported cryo-EM structure, complete structure of a B-cell antigen receptor published. And if it hadn't been for a couple of papers that came only 2 months ago on the cryo-EM structure of the human B-cell antigen receptor, this would probably be the first one in general. So this is murine BCR. And basically in this paper, I think it might be interesting to just tell you a little bit of what I learned from cryo-EM. How does this work, right? I was not very familiar with it. So basically what they did is that they generated a slightly modified B cell receptor in order to make it easier to properly isolate. So they have this. So every B cell, so a kind of a naive receptor for B cell is basically consisting of the heavy chains. In this case, if it's a naive B cell, it's going to be the IgGM, so the kind of the mu heavy chains, and two light chains, and an so kind of an IgA and IgB. They have these transmembrane proteins that interact and have the actual signaling domains in their cytosolic tails, so these ITAMs, and they are the ones doing the signaling. The idea is that upon antigen recognition, these motifs in the Ig alpha and Ig beta chains will be phosphorylated, and then kind of will continue downstream signaling down this receptor. And so basically, for this study, they generated an engineer BCR that had kind of the murine IgGM heavy chain, and a Ig alpha that was tagged on the one side with a flag epitope, and in the extracellular side, and on the intercellular side was marked with a YFP to kind of make it easier to identify. And then it was together with the Ig beta chain that had the actual item attached, which also is pretty neat. And they expressed this on a cell line, and then they purified it through a very difficult, must have been a lot of optimization, because these proteins, you need to generate them in a very they have to be really good quality. You must retain the structure in order to load it for the cryo-EM. And and as you, Jason, already know, but maybe I'll share it with our listeners. For cryo-EM, you get your very highly pure protein, and then you place it on a kind of a grid, right? And then these molecules, each of them kind of populates the grid, and they find themselves in different orientation, and then you freeze them in place. And so you end up having kind of thousands of copies of the molecule which you are analyzing, that is on the two-dimensional kind of plane. They are located, and you can see them in different kind of orientations. And then you can, if you take kind of cryo-EM pictures of each of these, you take the density of the signal that you get from the electron microscope, and then this together, you kind of, using, I would say, very advanced computations, you put them together to generate a 3D structure of your protein of interest. And that's basically what they did. So they used this. They had a data set of up to 400,000 particles, and they used that to bring to life a 3D structure of the B cell receptor. Interestingly, they also used AlphaFold to kind of generate a initial model to make it easier to then kind of fit it to what they see from their data. And I have to say that I did not think too much about the structure of the beta B cell receptor before, and there's a lot of detail about how, on the one hand, they could not really look into the structure very well of the kind of the FAB segment, which is the last kind of the very extracellular domain. The last two Ig domains are where the short chain is attached to, because it's very mobile, that area, because the hinge of the antibodies right before that, so that area moves a lot. Apparently, that makes it hard to look with cryo-EM. So actually, they ended up kind of taking that as from the crystal structure, which is designed to be very much easier to immobilize, and then they kind of attach that to the rest of the structure, which actually they could very well see with cryo-EM. So it seems to be quite a lot of steps necessary to generate this structure. And I guess that in general, this is a very important kind of kind of basic research to understand the structure, because it was not very clear, especially how the heavy chain interacted with the Ig alpha and the Ig beta, which are the ones that are doing the signaling. Interestingly enough, they show that although the molecule, kind of the extracellular domain, is fairly symmetrical, because it has like two heavy chains that are the same, and once it starts kind of wrapping on itself and going through the transmembrane domain, it becomes not so symmetrical, and then there's kind of a very specific way in which these peptides kind of are threaded with each other in a way that it seems to be very important to the whole structure. And I think when it comes to the what seems to be a very interesting observation is that there seems to be a confirmation of the resting TCR beta, B cell receptor, in which the item structure is kind of auto inhibited, suggesting that there might be a conformational change that happens upon binding to the antigen that's actually involved in the activation of the TCR, for the BCR itself. Because apparently, so far, the main mechanism was thought to be through kind of mostly clustering of many BCRs together, and then the items kind of activate each other. But apparently, the resting item is maybe not active on itself. So I think that if that proves to be true, that might be kind of a paradigm change in how we understand signaling through the BCR. So congratulations to the authors. This much has been a lot of work. And also congratulations to the people that published the previous papers that were those were published in Science in August. So they also, I guess, did a lot of work, and they had similar results with the human B cell receptor. Give or take some of the homology, which is not perfect, but the structure was also very similar in their hands.
I'm just going to state that structure dictates function as a biochemist and leave it there. But yeah, I think it's really awesome because now you're learning the structure, which means you can really learn how it functions.
Yeah.
All right. So I'm going to do the last little bit here, getting deep in the science. Next up, gut innervating nociceptors regulate the intestinal microbiota to promote tissue protection. STEMCELL, first author is Wen Zhang, last author is David Artis. It came out October 27th. So after now, it's early press released, I guess. But you know how it works. Time is a blur anyways.
Come from the future, Jason.
It's what it says on the bottom of it. All right. So nociception, pain signaling, right? In inflammatory bowel disease, it's known to be, painful. Lots of people feel with their guts, right? There's hypersensitivity syndromes, irritable bowel syndrome, IBS. They want to understand how the microbiota relates to this. So there's a certain set of nociceptors called TRPV1 nociceptors, which kind of are a generic nerve that innervates the gut for generic sensation. So unlike pain specific or heat specific, this is kind of a do-all. And what they find is that this receptor is critical in shaping the microbiota. And so they find that if you knock it out and give them DSS, colitis, the mice have worse colitis. And then they can abrogate this in different ways. They use DREADs, which are designer receptors exclusively activated by designer drugs. So these are GPCRs that are very specific for specific molecules that don't otherwise do anything, that you could put in, and they're G coupled to like G alpha I or G alpha S or GQ to have the opposite effect of your regular receptor, or the same effect. So you can turn it on and off in the same way and put it under the transcriptional control of the gene of interest so it shows up where you want it to show up. Actually, the person who invented this was Brian Roth, who was one of my committee members back at UNC way back in the day. So very, very familiar with DREADs. It's a big thing. So point being, they used a dread and showed that knocking it out was very similar to having a dread. They turned it off and then turning it on in the knockout remove the effect, right? Because if the dread's there, then it'll replace it. So they did this throughout the paper and showed that that lack of this innervation led to worse outcomes. Okay. And you can also do pharmacological ablation because there's a chemical that if you over signal kills the nerve. So they showed this like 3 or 4 different ways. And this chemical is RTX. Not surprising. It's a cell paper, right? So you're going to have 55 figures and 8 ways of doing it because they fully interrogate this, which is actually nice. So there's not really much doubt left. Then they show that knocking this out, if you get rid of this, and they chemically do it, but then showed other ways too. If you get rid of this, you have decreased Bacteroides and increased Firmicutes species. Okay. Then they know those are important for IBD in other papers and stuff. That was really interesting. They did FMTs and showed that you could take a mouse had this one of these knockouts, either genetic or chemically ablated or dreaded. You get this disrupted microbiota, you transplant it into a germ-free mouse, and they recapitulate the phenotype of worse disease. So then they're trying to figure out how this works, right? So they show that a few different ways. They did antibiotics ablation, showed that the microbiota responsible were vancomycin sensitive, gram-positives, and they get it down to it being a specific broad Clostridia consortia that's doing this. Then it's not related to Jackson mice versus taconic mice, because taconic mice are known to have these extra filamentous bacteria. It's not related to that. It's a very specific, it's not a couple of very common E. epicalum or Bacteroides, Theomicron. Point being, they did a couple specific cocktails. It was only a Clostridium species broadly defined that did it. They couldn't point it down to a specific bug, but it was vancomycin, sensitive Clostridia. Then they were like, well, how the heck is this working? So the other thing they notice is this neural pathway releases a whole bunch of substance P. And son of a gun, substance P is altered in this setting. You lose substance P if you knock it out or ablate it or dread it down, right? All the ways. And so they found then that the substance P from the neuron is actually what's driving the changes in the microbiota. So if they put substance P in the knockout, that substance P signals and causes the microbiota to go back to be normal or not. If you inhibit substance P, you have the disrupted microbiota, and it again transmits. So what you have is this crazy cascade. And I love this because most microbiome studies are not causative. When we talked to one of our guests previously, we're talking about like there's this chicken and egg thing, you co-evolved. How do you know what's first? In this case, neuronal signaling through nociception drives substance P, which shapes the microbiome in a way to be protective from intestinal injury. And if you lose that, you have a different microbiota that makes you more susceptible to intestinal injury. And they go down, as I said, the whole pathway showing that this carries all the way down.
I have a question. What exactly is substance P?
It's an inflammatory, like basically a cytokine.
Okay, that it's made by neurons.
Right.
Okay. And do they say how substance P interacts with the microbiome?
Nope.
Okay.
They just know that if they can give it, and it recovers.
Interesting. And these nociceptors, are they being triggered in any way, or is just
sure, by inflammation they're normally going to get triggered.
Okay.
Right.
So they pick up a little bit of inflammation there.
The point is, you can take a regular mouse and just the knockout on its own has a disrupted microbiome. Because there's nociception all the time, right? You're sensing stuff. So it's not just pain. That's just an exacerbated state, but it's sensing things all the time. Their microbiome is altered at baseline, and that shift in microbiome, if you transfer it to a healthy mouse and then injure it, propagates the problem.
Oh, okay. That part I missed.
But you're also going to propagate more signaling by the nerves during injury. And substance P is known to abrogate inflammation generally. and modulate it, but now this is really backlinking the whole thing.
Man, the more we think about it, this whole gut neuronal axis is terribly complicated.
Yep. It makes it
All
more
right.
fun.
Oh, for sure. For sure. We're never going to run out of papers like this to talk about.
No, we're not. Well, we did cover Gasdermin, and we're going to be speaking to Doctors Kristin Patrick and Robert Watson at Texas A&M in just a moment, where we get even more into Gasdermin, and
also mycobacteria. So got it all.
We do. But before we get to that, are you hungry, Brenda? Do you participate in the Immunology Journal Club and you wish there was better snacks?
Yes.
STEMCELL Technologies is offering you an opportunity to win $300 worth of refreshments, also known as snacks, to fuel your journal club discussions. To get the best out of your science and your journal clubs, and to find out more and enter the sponsorship contest, visit stemcell.com/journal club contest.
We are talking today to Kristin Patrick and Robert Watson. Dr. Patrick and Dr. Watson are assistant professor and associate professor at Texas A&M University. And very interestingly, they have a joint lab in which they do all kinds of research, including RNA binding proteins, macrophage activation, mycobacterium, tuberculosis studies, and also some joint projects, which are very interesting. We are so happy to talk to them today. Thank you very much for joining the Immunology Podcast.
I'm happy to be here.
Thanks for having us.
Welcome on board. All right, I'm going to hop in first because you guys have a new paper out in Cell, and this is one of your joint projects, I think, that talks about one of the molecules I got into in the last few years. I was a postdoc on Gasdermin D, and necroptosis, which is different than pyroposis, which is different than apoptosis, and all those, because we can't stop naming things in science. So can you guys give us a high-level overview on the story of mitochondria, Gasdermin D, and necroptosis, but also maybe the one sentence on what the heck Gasdermin D is, and not to be confused with A, B, or C?
So Gasdermin D is a pore-forming protein. It needs to be cleaved by caspases before it can be activated. So before it has that pore-forming ability. And it's typically studied in pyroptosis, as Jason said, where it forms holes in the membrane, and then that allows for release of cytokines like IL-1 beta and IL-18, and then also eventually allows swelling of the cell and lysis, leading to cell death. We found that Gasdermin D, in addition to making those pores in the plasma membrane, also makes pores in the mitochondria. And our work wasn't the first to show that, but it was the first to show that it led to a different type of cell death.
And we really got interested in understanding how Gasdermin D was regulated in the context of actually tuberculosis infection. And TB, one of the cell death pathways that TB induces is pyroptosis. But in the actual literature, there's a bunch of genome-wide association studies that have shown that genes associated with mitochondrial homeostasis require and induce susceptibility in humans to tuberculosis and other kind of mycobacterial infections. So taking it from the mitochondria, Gasdermin D, we found forms pores in the mitochondria. And there's actually this kind of gene involved in mitochondrial homeostasis called LRRK2. And for still unknown reasons, actually, LRRK2 drives increase in mitochondrial oxidative phosphorylation, which increases reactive oxygen species from the mitochondria due to overtaxation of the electron transport chain. And this combination of forming pores in the mitochondria, along with these differences in mitochondrial homeostasis, release abundance of ROS. And this ultimately causes a switch from Gasdermin D-mediated pyroptosis to RIP kinase 1 and 3 mediated necroptosis. And so we actually have the localization of Gasdermin D dictates kind of the kinds of cell death that occurs in these macrophages.
And how does this correlate to the infection itself? Is the mycobacteria somehow interfering or guiding this balance between pyroptosis and necroptosis?
Yeah, so the kinds of cell death that go on during tuberculosis infection really dictate kinds of the disease outcomes. And right, so generically speaking, this kind of non-inflammatory cell death called apoptosis is associated with kind of protective responses, immune responses during TB infection. And these more inflammatory kinds of cell death, pyroptosis and necroptosis especially, are associated with very poor disease outcomes. And so a mouse harboring this 1 point mutation in the gene called LRRK2, or this kinase called LRK2, which is a gain of function mutation, causes this switch and increased necroptosis. And this inflammation, TB loves this kind of inflammation. TB grows, and there's really severe pathogenesis of disease, increase of neutrophil flux, and severe inflammation. And so these mice succumb to infection really very quickly. It was just harboring 1 point mutation. So it's pretty striking.
And I guess I'll add on to that this is not, so far, seemingly not unique to TB, right? We saw this increase in cell death using other pathogens like Listeria and Mycobacterium marinum. So we kind of distilled it down to anything that activates the canonical inflammasome. In the background of this kind of this sensitized oxidative stress environment could trigger this kind of refunctionalization of Gasdermin D from a plasma membrane pore former to a mitochondrial membrane pore former and push this necroptotic cell death. So, while we focused on TB, we imagine and are sort of hoping that this will be relevant to other infections, right? And so we could potentially link this human mutation in LRRK2 or just oxidative stress in general to these poor disease outcomes based on kind of pushing this hyperinflammatory type of cell death that is something that we know the pathway, but the idea that it could be triggered in this way was new and potentially important.
I want to make a very kind of naive question. What is the best way of understanding the fact that if you have a pro-inflammatory cell death, sometimes you expect it's not activating the immune system and improving the immune response in a way, or because maybe I'm not very much of an expert on mycobacteria infections. So what exactly is the way in which mycobacteria harnesses this type of necroptosis to survive or it's just that the inflammation itself is detrimental for the host, which is of course terrible because if you're the host, that's you. But what's the balance here?
That's a very good question. So that's something that is observed in mouse models of infection of TB infection and as well as humans. So if you can recruit have this inflammatory cell death and you have like this influx of neutrophils, there seems to be a milieu that exists that TB loves to grow in, but that might be detrimental to other infections. So for instance, in this LRRK2 mutation, this is unpublished work actually, but we know that during TB infection in the lungs, there's massive inflammation and TB, the bacterial replicates really, really well. However, like in other kind of models of infection, such as Listeria, this inflammation seems to be protective against more severe disease and dissemination, the kind of disseminated disease during at least a mouse model of infection. So for whatever reason, TB kind of loves this kinds of inflammation. When you recruit excess of neutrophils, people have hypothesized that neutrophils may be a niche, which we can argue against that they're so short-lived that might not be the case. They might be protective in a way and a milieu in a way that TB can grow extracellularly. And so people don't quite understand, but anytime you get this influx of neutrophils and this inflammation, really it's been shown TB likes it. And this is a detrimental thing for the host and a positive thing for the bacteria to grow.
And this is the first time Gasdermin's been implicated in necroptosis, you said?
Yes.
I'm going to ask this only because I have knowledge you would never have seen because it was never published. How important is phospholipid signaling in this cascade as far as you know?
Oh, well, we're laughing because we are barking up this tree, I guess. So one of the major outstanding questions is, why is the N-terminus of Gasdermin D, the pore-forming portion, why is it localized to the mitochondria, right? So there's a lot of cool hypotheses that you can come up with, right? One is that there's some kind of change to the structure of the mitochondrial outer membrane that makes it look more like the plasma membrane, or exposes some other phospholipid that Gasdermin D can bind to. And we have some hypotheses about what kind of that remodeling could be, but we do think it's driven by this oxidative stress. So we're now getting into this idea that lipid oxidation and or the relocalization of certain phospholipids, perhaps from the inner mitochondrial membrane to the outer mitochondrial membrane, could be serving as a platform to recruit Gasdermin D preferentially to the mitochondria. But yeah, lipid, I'm laughing because for a long time I was like, I hate lipids. I don't want to study lipids. But you kind of don't get to pick what you study in biology if you follow biology enough. You kind of have to do it all. So yes, Jason, that's a really good point. We think that is an important player.
So I'll share this on air simply because I don't think it'd be fair not to. There's a family of proteins called the TIPE family, which is, we can get into acronyms later. They regulate phospholipid signaling and are intracellular transport of phospholipids. And if you knock out TIP0, the original one, Gasdermin D is massively overexpressed and its localization seems to be screwed up. And then the cells are wildly resistant to necroptosis.
Whoa.
And none of that got published because I ended up having to leave my postdoc because my PI left the States before the paper came out. And it's just sitting on my hard drive.
So, dear listeners, breaking news only on the Immunology Podcast, unpublished data from Jason.
Nice.
Well, it sounds like we're going to get a new collaborator here.
So we should talk. Other person that you might want to talk to is Igor Brodsky at Penn. He has really good C-tagged and N-terminal tag Gasdermins that we are doing localization with by flag protein, and we had that working before I managed.
We know Igor from our training. We all trained at Yale at the same time. So we can easily make that connection.
In fact, when I was talking about like visiting, I gave a seminar there recently. So
there you go. I was going to say, I know he has the goods. I didn't know if you've talked to him recently about his pile of constructs.
Yeah, he has a lot of good reagents in that lab, man.
All right. So now that we talked about what we're going to be talking about after we go off air. So you guys are unique in that you have a joint lab and individual interests. So before we dive into that whole apparatus and working together and the inevitable lab meeting conversations where you disagree with each other in front of the grad student, how do you guys each have your own interests as well. So could you guys maybe take a turn to describe some of your own interests, and then how you're able to, and then maybe after that we'll come into how you merge those and weave those together into a program.
Cool. Yeah, I can start. So my PhD is in microbiology, but I ended up doing my PhD in a molecular RNA biology lab. I studied RNA interference and African trypanosomes, which are still the best pathogen in the world, and they're my favorites. But I kind of got into RNA biology, frankly. I was really driven by kind of these molecular mechanistic questions. So my training was all in RNA biology. And when Robbie and I, Rob will tell you his path, but when he got recruited to Texas A&M, we sort of decided to work together for many reasons, which we can go into. But there was a clear opportunity for me to transition my interest in RNA biology to understanding regulation of the immune response. So that was a research program that I essentially created de novo when we started here, leveraging all of Rob's expertise and reagents, and now taking an RNA biology focused, an RNA binding protein specifically, focus on how are post-transcriptional steps in gene expression regulating innate immune outcomes, right? So we study signaling, we study transcription, we study transcription factors, we know all about NF-kappa B and IRF3. But what happens after those transcripts, like cytokines and chemokines? What happens after they're made? Are they processed differently in different scenarios? Is the timing of their processing important? What are the RNA binding proteins that are mediating these essential processing steps? This was something that was really sort of a black hole in immunology because so much focus had been on the signaling and the transcription. So I saw that space and decided my lab could fill that easily, kind of by combining our lab's interests. So that's the niche where we live in understanding how RNA binding proteins and RNA processing, like pre-mRNA splicing, how these processes influence innate immune outcomes in macrophages.
So I studied tuberculosis as a postdoc, and actually it was I studied enteric pathogens during my PhD. So I kind of stayed in the bacterial pathogenesis world, although thinking about, bacteria that cause diarrhea and a bacterium that causes a lifelong infection are a very different way of thinking. And of course, TB is a biosafety level 3. So that was like another layer of difficulty and difference between kind of bacterial pathogens. And so what YLab really studies is kind of the interface between the innate immune system and TB. And that's kind of our driving force. And so, TB is phagocytosis, and actually it's a kind of a first line of defense, usually these macrophages. And actually this is, it's evolved a variety of specific adaptations to kind of survive in this niche and replicate. And so I thought that was really cool. And these interactions are kind of where we start thinking about, and then we go from there basically. And so, TB is kind of thought to be a vacuolar pathogen traditionally. That means it kind of resides in this sealed membrane compartment. But we know now that it has a virulent secretion system, much like analogous to many other virulent secretion systems in gram-negative bacteria. And it actually creates holes in the vacuolar membrane. And what this does is this allows this interface of virus factors to the macrophage. And so there are generally 3 consequences. One is there's activation of a type 1 interferon response, which is at the transcriptional level. And so that's kind of where we started with Kristin's lab also, is we study the boring kind of signaling pathway leading to activation of transcription factors. Kind of the second major pathway that we've uncovered at that interface is a selective autophagy. So a population of TB are ubiquinated with an eat me signal. And this targets them to autophagosomes and destruction of lysosomes. And the third consequence that we ignored up until this last story is access to the cytosol, TB to the cytosol activates different kind of cell death pathways, including the inflammasome. And so really TB, 9 times out of 10, you don't even know that you have it. And so these pathways over the course of, coevolution for a very long time are very balanced. And we're interested in factors that kind of disrupt this balance between these different pathways. And so that's where kind of our interests lie, and my particular interests lie, kind of like studying regulation now of these pathways during infection.
So we have trypanosomes and TB, which are different a little bit. Infections and intracellular or innate immunity, some level of intracellular stuff going on. And then that's where you guys combine together. So you guys are both the same institution, you run a joint lab. How does that work? So my wife and I are both scientists. We've talked about having a joint lab together, but we definitely argue like an old married Jewish couple, because that's what we are at this point.
But not old. Come on.
I know. Well, we've been together for 20 years because we met the first week of undergrad. And so we've always been like, ooh, we already know what happened when we ran a martial arts school together and had a dispute. I can only imagine like lab meeting. So how do you guys make it work? And I say this because my wife in undergrad was in the Fulton lab at Brandeis, which was ran by a couple at the time. And so we've seen it before, and I've seen it in varieties, but it's tough. So tell us your secret to success.
Well, I also, back in my trypanosome days, also worked with a couple at Yale, Chris Chudy and Elisabetta Ulu. And we called them mom and dad behind their back. And when, mom was in a bad mood, we went and talked to dad. And when dad was giving us a hard time, we went and talked to mom. And, I think that as a student, as a trainee, having both those perspectives was actually really helpful, and having PIs that were good at different things really actually broadened my learning experience, and learn different ways to manage some of the things you're talking about. So yeah, I mean, Robbie and I spend more time together than most couples. Our kids definitely know words like TBK1 and LARC2 because we talk about science all the time. For the most part, we're try to be pretty professional at work. And even if we were in the middle of an argument at home, we kind of stop that and say, okay, I'm working on the AIMS page today. You're working on this today, right? And we kind of get in work mode. Our trainees might have a different perspective, but I think in general, we don't argue too much. Having slightly different backgrounds, I think helps, because there are definitely times where, I'll defer to Robbie on something TB related, and he'll defer to me on something transcriptional related, right? So the fact that we sort of have our intellectual supremacy, I think creates less stress as far as, us butting heads scientifically. So I think it works. What do you think, Rob?
I mean, of course I think it works. I think there are two kinds of people, ones that can work with their spouse and ones that can't. And I think I encounter people that's like, what do you do? How does that work? There's 0% chance I can work with my spouse. And so I am very fortunate in that we can. We've had a lot of experience working together in general. We met in graduate school, and Kristin's probably helped me more than I've helped her along the way, on anywhere from giving talks even in graduate school. And so we have a long history of informally collaborating, I would say, both at the PhD level. As a postdoc, we also did postdocs there was one lab in between us. And so we saw each other a lot and helped each other out along the way. And so it was kind of a natural thing. And Kristin's very good at everything. And I'm not very good at several things that she's very good at. And so I am very reliant on her. So for me, it's easy. I was like, otherwise I would be not near the scientist I am today without Kristin. So I think that reliance on that also helps out. But we do argue. The other day we did argue in front of everybody at the beginning of lab meeting. I caught ourselves and I looked around. Everybody's just staring at us. I'm just like, whoops.
How are they getting a divorce? Oh no!
From time to time when you can't help, does spill over.
I mean, I think it's wonderful. I mean, Jason seems so skeptical about the idea, but I'm pretty sure here they're making it work. So kudos to that.
Yeah, no, we've talked about having a biotech together now at some point versus an academic lab, but it's still, work then comes home with you in a whole other way, right?
It does. I think that our kids are heavily involved in soccer and other things that we tend to talk about outside. I mean, certainly it doesn't ever leave us, and especially with the advent of Slack, I would say that it's a 24-hour, a day kind of thing. But we have enough going on outside that it's just rushing around and trying to survive. And so I think that also helps us out a little bit, just having other activities and trying to take care of two children that are crazy.
But at the same time, we outline AIMS pages driving to travel soccer games, just so everyone kind of sees where we're at. That is our life.
Yeah.
That sounds efficient.
Yeah, we don't waste any time. One thing I will say that I think today, I feel like there is a lot of, to be a good academic scientist these days, I think involves a lot of training and, being a really good mentor to students, which is something that we're really passionate about. And I would say, I'm not sure I would have a ton of time for my students if I wasn't sharing some of the load. Right. And I think that, this allows me to be a good scientist, be on study section, go to meetings, but not have my students, be where's Kristin for 3 weeks, right? If they need a mentor, they can go to Robbie and get, as good, if not better advice. So, I think that this is something that maybe hasn't been the model, but maybe it should be. This is a hard job that requires a lot of work. And why wouldn't you want to share it with another equally, intelligent, supportive set of hands, right? We definitely do more than 2 times the work.
Right.
Together we can, it synergizes and we become more productive. And, at the same time, one of us is able to go get the kid with the 101 fever, right? So I think it's kind of a good model for academics in general. And I think it helps us be successful.
I guess that you don't have to be married in the outside life. You can also just have this with a particular Jason coined the term or used the term, how was it? Work spouse? That still seems like a smart strategy to get someone that has your back and share the good and the bad with someone. And it's always, I don't know, sounds like a good plan.
Yeah. There was a structural biology lab at Brandeis, biochemistry lab called the Petsko-Ringé lab. It's 2 people not married, had their own lives, but ran a joint lab together. And they were an example of a non-married science couple that did this. So it's interesting. I wonder, do you guys think this is going to become more and more the norm in some way with like either dual PI labs, couple or not, but as we go forward kind of with the ever shifting quicksand that is academic science these days?
I do. And I kind of told this to you guys before. I think that as I go around and tell how we go about doing things, people have like, oh, I like that model. And I think people are non-spouses, as you guys said, that are beginning to like this idea too. So I think it's become a more popular model. And I think it could be, I mean, you could do it almost like a married couple, or you could do it just like maybe like not as intensely, I guess. I think there's like probably more degrees to how much you collaborate or whatever, right? But there is a lot of, it doesn't like make sense on a lot of levels. Not only, I mean, even at the laboratory level, we're sharing, you don't have to buy 2 sets of restriction enzymes. I don't know if people use restriction enzymes, but like restriction enzymes or, Western blot rigs or whatever. Like, I think even like that, it's a more efficient way of running things on many levels.
Yeah.
And then also, I mean, with the stresses on funding, I think that we don't worry about that as much because we have 2 lines of money, basically 2 money streams coming in the lab, right? So if someone goes through a gap, then the other one can kind of pick up. And, that's what my postdoc, my PhD advisors did that. And I never was worried about any of that, but now I know the behind the scenes, like, oh, so-and-so like had a time where they didn't have their R01 and then the other one filled in. So, I think that if we want to try to create the most supportive, consistent training environment for young scientists, that to try to put this all on one person and have them be really supportive PIs, that are at kind of the cutting edge of research, I think it's hard to do everything. And then forget like throwing in a family. I mean, not even sharing a family, like if you have your own family, all of these stresses are, as we all know, right, we're coming to a breaking point. So I think the idea of being more collaborative in science, what does it matter if there's 2 names at the end of a paper with a star on it? Like, who cares? So, I think that's kind of where we're at. And we're hoping that, institutions start to appreciate that and don't care where the stars are.
For sure. That sounds like very inspiring, I have to say. I like that. I like that approach.
So before we wrap up, we always ask a fun question. So the question is, if you could not be scientists, but had to have a joint career together, it has to be 2 in the box doing something else. What would it be and why?
You could have different
absolutely
answers.
Can.
I have an idea. I think we could run a restaurant together.
Oh, that was my idea.
Married couple.
We're so boring.
Well, I think we both like food a lot, and we've kind of all kinds of different food, whether it's cheap food or and traveling and eating different kinds of things. So I think that was a natural place for my brain to go to. And I think also there are some deep parallels between, like chefs and cooks and running a restaurant and the science or whatever. So I think for all of those reasons, I think there's enough division of labor where you could be good at different things in the context of a restaurant and running a restaurant. I think that it would make sense, a lot of sense. So for all those reasons.
Oh, totally. The parallels are very clear. Like Robbie would be the one coming up with crazy new recipes that would require outsourcing some very annoying individual item. And then I would make like the classic tomato sauce that everyone liked, but it would like always be good and very consistent. So yeah, and that's kind of how our approach is to science too. But I think overall it'd be a great restaurant. You definitely want to eat there.
And I like the optimism. That sounds like a good plan. Thank you so much for joining us today. It was very interesting, and I really I enjoyed very much hearing about your perspective, and I do share your thoughts about the benefits of at least having another set of hands, an extra head to think along with you. And because it's a tricky job, right? Being a PI, it must be. I wouldn't know just now, but it sounds like a very tricky job, and it's good to know that you have someone to lean on. So congrats, and I wish you a lot of success in upcoming research.
Thank you for being on and sharing this pathway, and then of course the restaurant option at the end was a perfect cherry on top.
All right. Well, thanks for having us.
Thank you. It's super fun.
Well, that brings us to the end of our show, folks. Don't forget to subscribe to our newsletter at www.immunologypodcast.com. Hang out with us on Twitter at @immunopodcast. Email us at info@immunologypodcast.com. Send us some guests. Smash the like button on your podcast. Subscribe. Give us a review. Do all those awesome things. And we'll see you next time.